Hello everyone. Let's go ahead and start going through these genetics problems. We're going to take a look at the parents and predict what their offspring could be and produce genotypic and phenotypic ratios and all sorts of vocabulary. You'll see. So let's start with an example of the common garden pea that Mendel studied. So it says in Pisum sativum, the tall allele is dominant to the allele for short height. So you have a garden pea, and in this garden pea, tall is dominant to short. So a farmer crosses two heterozygous, hetero meaning different, so this is a different pair. A farmer crosses two heterozygous tall plants. So the plants are tall, but they're heterozygotes, which means that they carry two different alleles, which means even though they're tall, they carry the short. Then it says produce the genotypic and phenotypic ratios. Now, you don't know what those ratios are yet. It's no big deal. But let's go through and dissect this problem. This is very similar to math, where math really isn't about the numbers. Math is about procedure. These genetics problems are the same thing. Whether we're talking about humans or whether we're talking about birds or we're talking about the garden pea, it really doesn't matter. The genetics is all the same, and we dissect the problem the same way. So every genetics problem will start, and it will tell you about the organism that we're talking about, and then it will give you information about the alleles. So what do we know? We know that there's tall, and we know that there's short. And the problem has to tell you what is dominant and what's recessive. So in Pisum sativum, the tall allele is dominant, and the short allele is thus recessive. Then we have to tell you about the parents. So it says that the farmer crosses two heterozygous tall plants. This tells you about their genotype, what they carry in their genes. So that's step one. Whenever you start doing a genetics problem, the first step is to figure out what your alleles are, what's dominant and what's recessive. Once you've figured out what's dominant and what is recessive, you need to define these alleles. You need to pick a letter that represents the allele. You usually pick the letter based on the dominant allele. So since we're talking about tall, I'm going to use T. Some people use H for height. You could use whatever letter you want. It really is up to you. Just be aware to pick a letter where the capital and the lowercase, they look different. All right, so our big T allele is tall, which makes our little t short. So here are your two alleles, big T for tall and little t for short. This is the second step of doing a genetics problem. Okay, the first step is to read the problem and identify what's dominant and what is recessive. The second step is to then take those alleles and define them. Then we draw them out. So draw yourself a picture of a little plant, right? Plant, great. Here's your other plant. And what do we know about both of these plants? Well, let's take a look at their phenotype. Let's see what they are physically. So it says a farmer crosses two heterozygous tall plants. This tells us that both of the plants are tall. Okay, so what we're looking at then, this is their phenotype. This is what they are physically. This is their phenotype. They're both tall. Fine. Now let's take a look at their genotype. What do they carry in their genes? What do they carry in their DNA? So this is then going to be genotype. All right, so since the plants are tall, that means they are expressing a dominant tall allele. So since this plant is tall, it has to carry a big T. Since this plant is tall, it also has to carry and express a big T. But we know that all organisms carry their alleles in pairs, one from each biological parent. So there's a second allele here. Now, this could be a big T, dominant, or it could be a small t, recessive. Well, we know what this second allele is based on the problem. The problem says that these plants are heterozygous. They are a different pair. So this tells us that the alleles are different, which means that down here it is big T, little t. And then it says that both plants are heterozygous tall. Thus they are heterozygous tall, genotype, phenotype. So drawing out the parents and drawing out their phenotype and their genotype is the third step. So the first step is to read the problem, figure out what's dominant and what is recessive. Your second step is to define the alleles, and your third step is to draw out the parents. All right, now it's time to draw the Punnett Square, what Reginald Punnett gave us. And it's really a simple diagram. It's just a square. Now, even though we carry these alleles in pairs, we separate these two alleles. We don't give our offspring both. So this parent offers either the big T, tall, or the little t, short. So this parent contributes this allele here and this allele here. And then this parent does the same thing. This parent offers the dominant tall, and then this parent offers the recessive short. So this is what one biological parent offers, and this is what the other biological parent offers. So now these gametes are shared with each other. In plants, this is pollen in the air, sometimes with bugs like bees. And so this parent donates big T, this parent donates big T, and the offspring could receive big T, big T. That's the genotype. Or this parent could donate big T, this parent donates little t, and you get a heterozygote. Down on the bottom left, big T from one parent, little t from the other, you get big T, little t. Then over here you get little t here. These are the four possible genotypes of the offspring. This is step 4 of your genetics problem. So step 1, read the problem, figure out what your alleles are. Step 2, define the alleles. Step 3, draw out your parents, phenotype, genotype. Step 4, produce the Punnett Square. Now, step 5 is what the problem actually asked us to do. Produce the genotypic and phenotypic ratios. Okay, so all a ratio is a relationship between numbers. Oops, how about if we spell ratio with an A? There we go. So, genotypic ratio. We list all of the possible genotypes, right? So in this cross dealing with big T dominant tall and little t recessive short, the genotypes, what they carry in their genes, you always list the dominant first, big T, big T. You then list the heterozygote, big T, little t, and then the homozygous recessive, little t, little t. These are your 3 possibilities. You're not listing what's in the Punnett Square. When you make the genotypic ratio, you actually ignore the Punnett Square. These are what is possible with the alleles that are being presented. Now that you have your different possibilities, now you go back here and you look at what the possibilities of this cross are. So, how many big T do we have? There's only one. I would check it off so that you know you've counted it. All right, how many heterozygotes? You've got big T, little t, big T, little t. You have 2 heterozygotes, like such. And then how many homozygous recessives? You only have 1. So your genotypic ratio is 1 to 2 to 1. All right, phenotype. This is what these organisms are physically. So this is your phenotypic ratio. You only have 2 choices. There's only 2 phenotypes that we're talking about, tall or short, right? You always list the dominant phenotype first. So the dominant phenotype here, what they are physically, is tall, or the organism is short. Well, out of these 4 choices, how many individuals are tall? Well, any individual that carries and expresses the big T allele, the dominant tall, will be tall. So these individuals are tall. These individuals are also tall because they carry the big T. So both of these, even though they have a different genotype, even though genetically they are different, they have the same phenotype. They're both, actually all three of them, they're all tall. Then you go over here, and this individual with this little t is short. So while your genotypic ratio is 1 to 2 to 1, your phenotypic ratio is 3 to 1. When you then look at these individuals, since this individual is dominant, and they're the same, we refer to this individual as being homozygous dominant. Since you have a dominant and a recessive, we refer to this individual as a heterozygote. This one is also homozygous, but it's homozygous recessive. And this is then the last step of your genetics problem. Again, don't be distracted by the organisms, whether we're talking about birds or humans or pea plants. The genetics are all the same. It's a 5-step process to tackle any genetics problem. You read the problem and figure out who's dominant and who's recessive. You then pick a letter and you define your alleles. You then do a drawing and capture your phenotype and your genotype. You then come over here, draw your Punnett Square, and then you come down here and do your genotypic and phenotypic ratios. The procedure is the same regardless of the organism. Awesome, nicely done.